I hope you can hear me clear. And I would like to welcome everyone on this Early Career Members Network of the Metabolomics Society live webinar. This is the 16th series, 16th session, the last webinar of the year, which is hosted by me. My name is Aleš Kvasnička. I'm currently a researcher in Oslo University Hospital in Norway, and I am joined here by Loic.
Hello, so I'm Loic Mervant, and I'm currently a postdoc in London at the Crick Institute, and I'm doing mostly metabolomics and on cancer.
Perfect. So if you want to reach out to the EMN Early Career Members Network, you can do so on X or LinkedIn, or currently we also established a Blue Sky account, or of course by email if you have any questions afterwards, after the webinar, or just want to be in touch for upcoming webinars and podcasts and other things and events we do. But without further ado, I would like to now go to the most important part, of course, of this today's session, and that's our great speakers. So we are so happy to host here Dr. Daniel Petras and Dr. Paolo Stincone. And I would like just to introduce the first speaker of this webinar, and that's Daniel Petras, the assistant professor at the University of California, Riverside, and the University of Tuebingen. And Daniel Petras is a biochemist specializing in bioanalytical chemistry. He earned his diploma, master's degree in biotechnology from the University of Applied Science, Darmstadt, in 2012, and his doctoral degree in biochemistry from the Technical University Berlin in 2016. And after postdoctoral work at the UC San Diego, where he focused on environmental metabolomics, he founded the Functional Metabolomics Lab as an independent junior research group leader at the University of Tuebingen in 2021. And in 2024, he and his group moved to the UCR, where he holds he was appointed as assistant professor of biochemistry. And his research centers on developing mass spectrometry-based techniques to study chemical interactions within the microbial communities.
Great, so I'm glad to introduce the second speaker of today, so Dr. Paolo Stincone. So Paolo holds a PhD from the University of Rio Grande do Sul in Brazil, and is now a Marie Curie postdoc since 2021 at the University of Tuebingen. And before that, Paolo did his education in Italy, and notably in the University of Palermo and also the University of La Laguna. And yeah, we're very glad to welcome you two. And yeah, now time for the show, and the floor is yours, so go ahead.
Basically, I will just mention that the topic of today's webinar is a functional metabolomics toolbox for uncovering the small molecule dynamics in the microbial communities. And just for a technical note, I will mention that the questions, you can drop the questions in the Q&A box, and then these questions will be answered at the end of the webinar. So please stay focused until the end, and we will focus on the Q&A session at the end. But without further ado, I will, as Loic already said, we will leave the floor to our esteemed speakers.
Awesome. Well, first of all, thank you so much for the very kind introduction, and yeah, for like the invitation and giving us the opportunity to present a little bit of our work. This is, I think, a very awesome setup that you're having, because it's not only giving like, you know, like a slot for like the PIs, but also for like the people in the lab that do most of the work. And I'm very grateful for Paolo to be here as well and show some of the findings that we made. And I think this is quite fitting, because while we're primarily a tool development lab, there's some very specific biological questions we're after. And Paolo has been there since basically like the very first days, as he was like the first person I hired when we started in Tuebingen. And yeah, he's been seeing the tool development and then applied those to his biological system. And I'm very happy that he will showcase some of that. But yeah, before we get to that, I want to talk a little bit about the functional metabolomics tools that we're developing in my lab, and what the direction is. Before I get into it, I want to point out that this has been work by many people. Some of them are listed here. So my first slide is already my acknowledgement slide, and I want to point out that this would have been impossible with like the different lab and many different people in all of these labs that contributed to that. So yeah, overall, what we're interested in my group is basically to figure out what the role of like metabolites and particularly specialized metabolites is in structuring microbial communities and how that translates to like host and ecosystem function in the big picture. And yeah, I want to start with like one molecule that is very special to me because I spent a lot of time with that molecule during my PhD thesis in the group of Roderick Sussmuth at a technical university in Berlin, and that is albecidin. So albecidin is a non-ribosomal peptide with a fused polyketide moiety, and it's exclusively made of this non-canonical amino acids. Right, so it's made by a non-ribosomal peptide synthetase, and in a fairly complicated manner is assembled. And this is being quite an important molecule because it's considered the main virulence factor for the sugarcane leaf scald disease caused by a bacteria, Xanthomonas albilineans, and that used to be the causal agent for massive crop losses in sugarcane plantations. So now you can see here on the bottom right, there's this like white like lines. This is basically caused through chlorosis. And yeah, like albecidin is the molecule that is made by Xanthomonas albilineans that causes like this chlorosis. Now, what made it very interesting for us was that besides inhibiting like the topoisomerase of like the sugarcane, it also inhibits like the DNA gyrase of gram-positive as well as gram-negative bacteria, and it makes it like an exceptionally good antibiotic in like the nanomolar range. So now this is an interesting new lead structure for the development of new antibiotics, which are urgently needed. But yeah, what made it very special for me, again, is like I spent a lot of time actually reconstituting the biosynthesis by purifying many of like its biosynthesis genes and then building up step for step the biosynthesis in vitro. And that involved a lot of like protein purification and in vitro assays, including mass spectrometry assays, to basically figure out how albecidin is made, so we could come up with like a pathway. So something you till that point definitely did not find in Keck, and I think there's many such pathways that remain there. But yeah, the main take-home message from this time for me was that first of all, it took me really long to purify all the proteins to like figure out the biosynthesis. But also before me, many PhD students spent their time culturing hundreds and hundreds of liters of Xanthomonas albilineans cell culture just to purify the couple of milligrams that at the end were needed to like figure out the structure in the first place. And that was really a monumental work, and like it took a lot of time and a lot of money. So now the question, of course, is why is there such like minor amounts only being made in cell culture? And it was at the very end of my PhD that my boss at that time allowed me to go out for a visit to Montpellier, to the lab of Stéphane and Monique at the CIRAD Plant Institute, to actually work with like some infected sugarcane plants where we directly isolated albecidin out of, and then like quantified this in first like a bioactivity way here on simple like agar plates, but then also with a triple quad assay to then just figure out that actually in its natural system, as you can already estimate from some of like the massive halos we saw from the crude extract, is produced in much higher amounts. As well, around the same time, when we published the structure and the biosynthesis, very interestingly, the group of Rolf Müller, as well as a group in Korea, published very similar molecules, cystobactamide and coralomycin. And now, by looking at the structure and this very unusual para-aminobenzoic acid moieties, you could probably guess that there is a very high homology. And by looking at the biosynthetic gene cluster afterwards, it was very apparent that this must have like a very similar origin, right? So now cystobactermide and corollamycin are both made by myxobacteria, whereas albicidin is made by Xanthomonas, right? So like taxonomically like very far away. So question is, why did Xanthomonas like take up like that biosynthesis gene cluster? And how did that happen, right? And with those kind of like open questions after 3.5 years of PhD studies, it really left me with like the, I don't know, more question marks. So why is that like produced in such small amounts? Why is it produced in higher amounts? How is it transferred and why? So really, what is it that like, what is like the real role in like the environment? Why is it like made in the first place? And how does the environment and like, all like the microbiome around it actually contribute to like the production of these molecules? And yeah, with those questions in mind, and joining Peter Dorstein at the University of California in San Diego, like I really dedicated the next years of like developing large scale metabolomics tools for basically detecting specialized metabolites in ultra complex samples. And I think as most of you here know, mass spectrometry is an excellent tool to measure like a huge variety of different molecules and ultra complex samples. And yeah, we can measure them very precisely and calculate molecular formulas. But then, of course, this is not enough to like determine what the actual molecule is. So we go a step farther and acquire a lot of like MS-MS spectra. So now, and this to me at that point was kind of like the groundbreaking work that was done in Peter's lab, and why I really wanted to go to California and learn it, was what are we going to do with like this large amounts of data? Because this is, for me as a PhD student, was kind of like not really capturable. So I was like really intrigued by the molecular networking idea that came out at that time, and I was really happy to be then in Peter's lab to contribute to the further development. So in case you're not familiar with it, what we basically do is we sort spectra by pairwise alignments, and then structure like the chemical space within like this complex data sets, right? And if we can then annotate one molecule, we can like simply propagate annotation throughout these networks. And the idea is that when we align the spectra, we look for like similar patterns of peaks that are shifted by the delta of exactly the difference between the parent mass, right? And that allows us to basically connect molecules that are not identical but have highly similar patterns, such as here in this example where there's a methylation occurring, at the molecule at the bottom. And then we would assume that in addition to the precursor mass that is shifted by the delta of the methylation 14, also some of like the fragment peaks are shifted. So now over the years, we expanded this also to not only look at like MS2 data, but together with Robin Schmidt, we integrated some correlation analysis to also basically annotate all other ion adducts as well as like in-source fragments that frequently occur, to then structure them in a like a more sensitive molecular network, which we call ion identity molecular network. So now, yeah, over the years, this has really, I think, developed and has come a very long way. And I'm very glad that we could contribute to some of those developments. So what my group now heavily relies on is like, non-targeted LC-MS-MS with still data-dependent acquisition mode. And then, we use MZMine for feature finding. And then now GMPS2 and Sirius mainly for annotation. And now we developed our own little stats pipeline that directly plugs into that analysis. And now I assume many of you here are familiar with some of those tools and have mastered its use. And you probably all know that it's not straightforward, and that this data analysis part is perhaps like the most challenging endeavor of the entire experiment. And learning that, at least that's what we see when people come new to the lab, is typically what people struggle most with. So yeah, it's been really a pleasure to work with like many of the developers, including Ming, Tomasz, Steffen and Robin, but also Axel and Abser at the University of Tübingen, to kind of like streamline these pipelines and these different tools, make sure that they interact well, but also ultra simplify some of these analysis. And yeah, I'm particularly happy that now, you know, within like simple web applications within your web browser, you can technically do all of like this analysis just like with a couple of clicks. And as many of the you know, data is in the cloud and all the tools are in the cloud, it's really like no downloading, no installation involved, and you can like fairly quickly generate such molecular networks and analyze your data. So now, yeah, here, if you're interested, I put down the URLs for some of those tools, so please check it out. And also, in case you're interested in learning more about EMSImine, we currently have a seminar series going on through like the VMOL seminar series where Robin, Steffen, Ansgar, and Corinna are actually showcasing the latest developments of EMSImine. But now, yeah, so armed with this non-targeted metabolomics tools and data analysis pipeline, you know, we applied this to various biological systems, ranging from classic natural product discovery, is where I'm mainly coming from, but also to plant microbe interactions, to like human host microbiome interactions, and then perhaps what I have now closest to my heart, environmental systems, and in particular marine metabolomics questions. And yeah, in a nutshell, what we can achieve from there is if we combine yeah, like the non-target metabolomics with other omics approaches such as like metabarcoding or shotgun metagenomics, we can start getting like a fairly comprehensive picture of like microbial systems, right? So here, for example, we went to the California current ecosystem. This is a upwelling system in the Pacific here off the coast of California, where we sampled over many days water samples to map the interaction between like the ocean metabolome and the microbial community. And yeah, what we can do is we can structure like the microbiome here, for example, around Glucococcus. This is a cyanobacteria that is like very high abundant in the open ocean. And yeah, we can figure out what other microbes are around Prochlorococcus there, right? Through like a correlation network. And then in combination with our non-target metabolomics workflow, we can map the chemistry among like those communities, and from there build many, many hypotheses, right? And here, one example that I found intriguing and that popped up to my eye immediately was that some of the heterotrophic bacteria surrounding Prochlorococcus are likely producing homoserine lactones, here the molecule at the bottom, which is a known signaling molecule, right? Like involved in quorum sensing. So now there's, of course, like a very big caveat in that. So first of all, like the limited annotations of metabolites. So we're still here probably below 10% annotation rate. So 90% of the features we have, we don't have like level 2 annotations for it. And then even if we have them, we don't really know what like the molecules do unless there's like a paper describing quorum sensing, right? And in the case of homoserine lactone, this is an obvious one because it's like very widely known in microbiology. But yeah, like what about like all the others, right? That we either don't know the structure or we don't know the function. So really what, after 5 years of postdoc, this left me with was that like we can map like the chemistry and like this complex systems, but it's really like this dark matter of like the metabolome, or in the ocean of the metabolome, however you want to call it. And yeah, it was like this question in mind, and or was this I don't know, frustration in mind and overwhelming feeling in mind. I thought, well, how about we start like developing, you know, maybe some new hardware tools to, in addition to like identity, also add directly some functional information to that. And yeah, with that, I just want to point out that again, like when we think about like, you know, like a biological system and chemical space, and we want to use non-targeted metabolomics, which I think is a great tool, we typically go from like a chemotype here in the middle, right? Then through our non-target metabolomics pipeline. And at the end, what we get is a very long inventory of like all molecules that we can detect. We describe them as features. But then what is like really needed is like the stats and like the downstream analysis and verification to create like this inventories in some biological insights, right? And I think this is besides like learning all these tools, what I think is like the key bottleneck, and where we spend months and months, if not years, to actually follow up on like one individual data set. So now at the same time, there's new tools emerging from various labs, right? And that try to aim to like provide functional insights directly to such a study, right? Simpler experiments could be like a I don't know, correlation analysis with some other omics studies, right? Or like I don't know, like genome-wide knockouts with like CRISPR, also to then get like some correlation-based insights. But there's also tools available that give you actually some causative information, where you can directly get some mechanistic insights to the to that data set. So now, yeah, with that in mind, so this is, I guess, in a nutshell, what we're trying to do now in my lab. So we combine non-targeted metabolomics and sequencing to then, on the fly, add additional assays or technologies to get like some like functional information to that complex data sets. And yeah, just to highlight a couple, so there's different routes we're going. We're working with like bioactivity, we're working with native mass spectrometry, and we're also working with like chemical metabolomics to like infer reactivities. And as a large component of the lab, we're working a lot with synthetic microbial communities as a kind of like model system that actually allows us to like also like speed up like hypothesis testing. So now I don't want to talk so much about that, because that is what Paolo is going to talk in cover in his talk afterwards. But for the remainder of the time, I want to quickly mention like our bioactivity approaches and native metabolomics approaches. So yeah, in bioactivity-based metabolomics, it's kind of like a very old thing, because bioactivity-guided natural product discovery has been around for many decades, where you typically purify some molecules out of a culture. Here, for example, our Xanthomonas albilineans, and then extract it and then separate it over like a chromatography dimension, and then make like some bioactivity assays, right? So here in the simplest form, we could do some halo assays with a reporter strain, and then figure out, okay, which of the fractions are actually active, and then go after them and figure out what molecules are present in there. So in our case, here, this was some work I did in Peter's lab with like the strain I brought from Berlin. We figured out, okay, there are some fractions that have like high bioactivity, not only the main albicidin we already knew, but also some others. So we then took a deeper dive, with like the non-target metabolomics tools of specifically these bioactive fractions to then figure out what else is there. And yeah, in addition to like the main albicidin that I showed you at the very beginning of the talk, thanks to like the molecular networking, and then also later on now, thanks to like tailored spectral queries with MassQL, we could quickly figure out that, oh, actually, in addition to like that one main albicidin form, there is a bunch of like other derivatives present, that are all like structurally similar and all bioactive, some of which were even more bioactive than the main albicidin. So yeah, this is a very powerful tool. But the hook here was that like we knew already that there was like one bioactive molecule, and we kind of knew the structure. So, with the networking and MassQL, expanding that chemical space was fairly simple. But like identifying something like completely new, where you don't have like any seed annotation, that is still hard. So now, when you think about the bioactivity approach, right, what we of course hope for is, if we want to identify the component in a complex mixture that is active, we want to separate them over chromatography and then perform of our different fractions bioactivity screens. And ideally, we have such a scenario here where there is a first fraction here at the beginning, then there's a fraction in the middle and one at the end, and the one in the middle is active, whereas the flanking ones don't show activity, right? And then there is one peak in that middle fraction, so we can say, well, that is our bioactive molecule. So now, unfortunately, reality in most extracts looks rather like this, or much, much worse, right? So we still have a lot of like overlap, coelution, and like the fractions are like very rarely only containing one molecule. So like, while we can like, shrink down the number of possible candidates, it's still really hard to pinpoint exactly like the bioactive component. So now this is a long-term idea we developed. What we thought about was, okay, now that is of course like a problem, but also typical fractionation, you slice your sample into like, I don't know, like minutes or like 30-second windows due to like the typical configurations of the fraction collectors that are available. But what if we had a hardware that would allow us to make those windows very, very small? So small that we could like sample bioactivity at actually the same frequency as we sample the mass spectra, right? Which is typically duty cycles around like 1 Hz, right? So then we could actually not only, look at like this broader windows, but we could look at individual data points and then correlate those data points against each other between bioactivity and the MS signal to then prioritize like the best co-eluding peaks or like information, between the bioactivity signals and mass spec. And then, like kind of like distinguish between overlapping features, whether they're like, better or worse correlating. So now, that hardware unfortunately did not exist out of the shelf. So we spent quite some time building something ourselves, and it was really a pleasure to have Albert Hernandez here joining the group also at the very beginning. So Albert is a mechanical engineer who I met during my postdoc time in San Diego, who was adventurous enough to join us for 1 year in Tuebingen to gain some work experience abroad. And Albert's main project was to like build like a micro spotter that would be capable of spotting at high speed with like faster than 1 Hz spotting speed. And we bought like here this kit to like build like a CNC robot, typically used for DIY milling projects. And after a little bit of puzzling and construction, after a couple of weeks, we actually managed to like build here like the spotting device in orange next to the LC system. And what this micro spotter or micro typer can do is like, it's very simple, right? Like after the HPLC column, we have like a T-splitter, and then half of the flow from like the HPLC goes into like the spotting device here on the left, and the other half goes into the mass spec. So the capillaries are more or less like the same length, so that we have the same delay times and don't have to worry so much about synchronization of the data streams. But like roughly we get split signal coming from the microspotter and split signal from the mass spec. So now a really key element here was that when we have now this microspotting devices for which we use like a microfluidic device, we need, of course, like to get like a readout. And here it was really critical that we worked together with the group of Heike Protz-Esterheld in Tübingen, who had like bioreporter strains that up on specific antimicrobial activity, they emit like some luminescent light, right? So our approach works following. We take the microfluidic device out of the spotter, and then after the analysis, we overlay it with like agar and a reporter strain, and then simply measure like the luminescent light that is emitted if there is some bioactive compounds present. And then we convert this into like a bioactivity chromatogram, and then like perform correlation analysis between the two to then pinpoint exactly those peaks that are actually bioactive. And in case you're curious how this looks, so this here is the spotter in action, where you can see at around like 1 Hz, like it moves over our microfluidic device, and then it deposits individual droplets. So it's really just like a little bit of mobile phase that soaks into like the paper-based microfluidics, and then quickly evaporates. And here, as you can see now, some blue dye is eluting, and at the bottom here from like our detector signal, you can see nicely that it matches well with like the chromatography. So now, yeah, building this first prototype to actually using it and flushing it out so that it becomes some discovery. This was the next critical step. And here it's important to mention Christian Geibel in the lab, who was a postdoc coming over from Michael Lemmerhofer's lab in Tübingen after he graduated, to really bring like this microspotter to like an application stage. And he worked closely with Julian Schubert from Heike Protz-Österheld's group, who did like in parallel like all like the microbiology work. So this was really a great team effort, and I'm very thankful for them to have pushed it that far. And what you can see here on this like slides is simply we took like a bunch of like different antibiotics for which we had like different reporter strains, and then like basically spotted them on our microfluidic device, and then you can see nicely that there is like this bioluminescence signal for like the different compounds indicated as like these rings. This comes because like at the specific concentrations here, we had already some halos forming, so there's no growth anymore in the middle because it's really active, right? And we just like see the signal at the edge of the halo. But when the concentration is lower, you would simply like see like a black spot here, like you can see on the right. So yeah, apparently like the technology worked, right? So we had here some ground truths, and we could like nicely evaluate this. We could take some known authentic standards, perform like dilution series to like determine, limit of detection, which was here like in the nanogram range, but also like figure out what like dynamic ranges we can use, so whether we have to like dilute some samples or concentrate some samples to cover wider concentration ranges. So now, of course, like that creates also like a lot of data and like these images. And there, the other component was, what are we going to do with the data afterwards? And here I'm very grateful for Simon Knoblauch, who was a master student who, during a very short time for his master rotation project, actually developed a Python tool in form of a Streamlit web app called Microspot Reader, where we can actually do densitometric analysis. So we can like take like an image like this one here on the left, put it through like the software, and then it basically like recognizes the spots of like bioluminescence, measures like the diameter, as well as like overall signal intensity. And then like the very simple tasks of like the microspot reader is to like convert this into like a numeric value, which we could then like here display in a heat map, but more importantly, convert into like a chromatogram, which we can then downstream correlate against like the extracted ion chromatograms from the mass spec data. And then with like the mass spec data stream in hand and the MS data, from there we can use our standard molecular networking pipeline to actually then assign and dereplicate like putative like IDs, and if they're unknown and new, then go the step farther and purify the compound for additional structure elucidation and validation experiments. So now, how does that look with a complex sample? So here we took like some crude extracts my colleagues had like sitting around in Tübingen, and we performed like the microspotting. On the left you can see like the total ion chromatogram, so we can see that there is indeed like a lot of like different molecules eluting. And then when we now looked at like the bioactivity readout, just like at the microfluidic device after bioluminescence reading, we can see already, there is like a peak here on the red on the right, like red circled, but also there are some other smaller ones here. I don't know if you can see my mouse. Here on the middle of like the other plate, right? So I don't know what that is, but anyway, now pushing this through our pipeline, we could like rapidly then generate like a bioactivity chromatogram for the peak here on the right, and then overlay this with our LCMS data to then figure out fairly quickly that, this is actually erythromycin, which was like not so surprising because like the Saccharopolyspora eritrea was a known erythromycin producer, right? So it's kind of like also like a positive control. Yet, like the other signal we detected, which had a different retention time, here on the left, I mentioned before, this we could not dereplicate. So there, we could now go a step further and purify that and check out whether this is actually another derivative or even like a new structure that is bioactive. But yeah, so with this, we want to now, of course, like apply this to more biological systems. But one has also to mention that still the speed and scale at which we can perform this does not fully match non-target metabolomics throughput, right? So typically in large-scale experiments, we would measure hundreds to thousands of samples. So now dealing with like thousands of like microfluidic devices and doing like the readout, that might be challenging, right? So here, I really want to shift gears like a little bit and quickly mention our native metabolomics approach, because I think this has, in terms of scalability, maybe other capabilities that could speed up like that process a little bit. So yeah, native mass spectrometry has been around since like a long time, and especially in like protein mass spectrometry, this has been massively used, and it's a huge field. Where people use mass spectrometry to get like structural information about proteins, right? And the basic idea is that you ionize your protein at like a pseudo native pH and buffer condition to maintain its fold, right? And then you can like, study, I don't know, like stoichiometry between like, I don't know, multimers, or you can also study protein-protein interaction. So you can really maintain non-covalent interaction in the gas phase, and that works for proteins, but it also works for proteins and small molecules, right? So this is really an intriguing tool that has been widely used in the field to figure out what metabolites could bind to like proteins. So why can't we use that in metabolomics, right? To like add this as a additional activity information. So here, the problem is that typically we do not have like native conditions at all, right? We use like acetonitrile as a mobile phase, typically formic acid as a modifier, so all not very native at all. So to bypass that problem, Allegra Aaron, back then also a postdoc in the Dorstein lab, so she and I came up with like this concept that we can modulate pH after the column to then like infuse binding partners, and on the fly, in addition to just like identify molecules, we can also gain some sort of like binding information. So Allegra was mainly interested in siderophores and metals. So for the work with her, we mainly infused post-column then metals. But since then, we also expanded this to make use of this native metabolomics concept to look for protein small molecule interaction. And yeah, as I mentioned, the idea is very simple. We have here like a peak splitter that after like our separation, after the column, we simply infuse like a buffer, typically ammonium acetate, to like lower down the acetonitrile concentration and increase like the pH to like some pseudo-native conditions before we then infuse a binding partner. So either metals or now here proteins in that case. And then if like a small molecule binds, we would then be able to measure a shift in the mass of the intact protein. So now, yeah, how does that look? So the setup is fairly simple. This is our system in Tuebingen. And when we zoom in here, you can see simply after the column here on the right in the column compartment, there's like two T-splitters. The first one connects to like a makeup pump, and then the second one here connects to like the syringe pump from which we infuse like the protein, right? So like super simple design. If you have a secondary pump sitting around somewhere, it's easy to build. The only thing that we engineered was like here a custom fridge for our syringe pump, because some of the proteins we work with, they don't like room temperature so much, so we like to like cool them while they're still in the syringe. But for many proteins, you also don't need that because they like room temperature, like trypsin or so this is not a problem. So yeah, basically to build a very simple hardware setup, you need, of course, your LC-MS-MS system, then a secondary HPLC pump for makeup, a syringe pump that you typically have for calibration, and then 2 peak T-splitters and a little bit of extra tubing. So not much at all. And yeah, with that in hand, you can technically perform native metabolomics experiments, where again, we would assume that when a small molecule elutes from our chromatography column and sees like the protein, then it would bind, and then we would observe like a shift in the binding in the mass of like the apoprotein, right? And that would be specific to a given retention time at which like the small molecule elutes, and that we could then observe like a drop in the like blue signal here. So this is the apomass of the intact protein, and then like a new mass would appear at exactly that retention time here, when like the small molecule and the protein shift. So now, yeah, with this in hand, of course, we can then also turn off the protein infusion and just do like a regular metabolomics experiment with the same gradient. So we can use the same retention times for then matching of our classic metabolomics data with like the native metabolomics data. And then again, use some correlation analysis to match the 2 data streams to then, yeah, like create like this protein metabolite interaction network. So now, yeah, with this idea, we wanted to test it. And as a first system, we used here streptavidin, which is a known very good binder. And then, yeah, we simply infused that like while we separated some Pseudonychia cell extract. And as you can see here, the intact protein signal, when you turn on the pump at the start signal, then the signal comes up. Then in the middle, it drops. So there's this dip, and then it comes up again. And then, yeah, like it continues till we then stop the infusion and the signal drops again. So now the question is, of course, what's happening in that window here in the middle? And then when we now simply take a mass spectra at this retention time, we will see that actually here our multiple charged species from like the intact protein, they're shifted by a mass, and that when we now create an XLC from this new mass fits in perfectly into that window, right? So exactly what I showed you on the last slide. Now, question is, of course, what small molecule falls into that window and what is the mass, right? So here, when we deconvolute the multiple charge species, we get a mass difference of roughly 977. Keep in mind that this is like not super accurate. So there is like a couple of Dalton or Thomson mass inaccuracy. But nevertheless, the retention time fitted here very well to like a small molecule, right, in red. And now we can look what the mass of this is. And this was 244. So that, of course, does not match 977, but it won't take you long to like realize that you have to multiply this by 4 to like reach a very similar mass than that. And then we ID'd it, of course, and we also knew already what is a very good binders for streptavidin. So this is biotin, and biotin, of course, has 4 binding sites on streptavidin, so it fits perfectly with like the literature knowledge, and showed us nicely that in theory, like this approach, at least for a very strong binder, such as like streptavidin biotin, should work. So now, yeah, armed with like this new tool, we sought out to like screen for some new, like inhibitors. And at that time, I was very interested in cyanobacteria because of like some of the field work we did and some of the data we already had. But most importantly, because Raphael Rare came to our lab after a talk, and he said, "Hey, I have like this extracts from like this biofilms from corals, which I think contain a lot of like Rivolaria cyanobacteria, and I think there might be some protease inhibitors in there. So couldn't we screen them?" So then we simply like took like the samples from him, extracted them, and then run them over our native metabolomics setup with like a protease, in our case chymotrypsin, as a target. So now here for chymotrypsin, this very same kind of like overall signal structure appears, right? We get like an intact protein peak, or peaks from like the multiple charge pattern that then, you know, like goes here over time. Then we have like a couple of dips, and then, you know, at the end comes back up again. So now our question, of course, is what is happening within like those dips, right? And here again, we see very nicely in our metabolomics data that there is some peaks that fit like nicely in there. And when we now look at the masses and the mass differences between the apo and holo, we see that we have a delta of 1155 Dalton, right? So we know, okay, maybe that is the binder. And when we look at the metabolomics data on the right, then we will see nicely there is a peak with 1156, which we think is the M plus H plus, as well as like here an M plus ammonia, as well as like some water loss in source fragments. But like if we would now calculate the neutral mass, it would be 1155, right? So this is great because that means that, well, apparently we have evidence that there is a putative binding event between like this 1155 metabolite and chymotrypsin. So now that's great, but really interesting, it became when we deconvoluted all the data and then started to like go through like the entire chromatogram systematically, because what you can now nicely see is while there is here our apo peak around 25 kilodalton, so again, this is deconvoluted, we also see every couple of seconds that there is some new peaks emerging in the higher mass range, right? So indicating that there is not only the 1155, but there is actually a couple of other species that might also bind chymotrypsin. So now with our computational pipeline in place, we can like create extracted ion chromatograms from like all of these new emerging complexes to then, you know, like correlate them against like some of the metabolite data. And then now in combination with molecular networking, we can build like this interaction network where we can now show, well, there is interaction between chymotrypsin and some of those features indicated through like the red dashed line. And then a lot of like those binding features have also high spectral similarity between each other, indicated through like the gray edge. So now that is, of course, very exciting because it means, well, there is an entire family of like putative protease binders, right? And now we have a target. And thanks to the hard work of Raphael, we could then purify some of them to then, first of all, elucidate their structure through a combination of MS-MS and NMR experiments. But then most importantly, with the pure compounds in hand, we could do like some follow-up experiments where we did titration experiments to determine LOD, but also get an estimate on binding affinities. And perhaps most importantly, with the lab of Anthony O'Donoghue, take those compounds and use other established bioassays to actually determine whether, like here, the Rivolaria peptides are indeed protease inhibitors, which turned out to be indeed like nanomolar active for a bunch of like different proteases. So now, yeah, with this, we discovered a new family of, yeah, I think strong protease inhibitors. And we can now, of course, like expand this to other proteins. And since then, we've been working with quite a couple of different targets. Due to time, I don't want to go into all of them, but I hope that I could kind of like highlight a little bit how some of those tools that we develop like help to structure better like the molecular function of metabolites and help us to better understand microbial communities and ecosystem, and hopefully shed a little bit light on the dark matter of the metabolome. So again, this was work of many people over multiple institutions. So I'm very thankful for my former advisors, Peter and Lihini at UC San Diego and Scripps Oceanography, and then also my wonderful colleagues in Tuebingen and now in Riverside, and perhaps most importantly, all the great people in the lab are present and former lab members who are like super fun to work with. And yeah, again, thanks for having me here and giving me the chance to show some of this.
Fantastic, Daniel. Thank you so much. It's a fascinating lecture. I have many points and many questions, also some in the audience. But until we get to the questions and answers, which you can, just information for the attendees, you can drop that in the Q&A session. But just before we jump there, of course, there's the second presentation from Paolo Stincone. So floor is yours. You can share your screen and
Yeah,
we'll just continue.
thank you. Okay, you can see my slide, I believe.
Yes, looks perfect.
Yes.
Thanks.
So can I start? First of all, thanks for the invitation, and it's a pleasure for me present here some of my data. This is our data coming from a project that from which I actually got Marie Curie individual postdoc fellowship. And the title is Elucidating the Role of Microbial Metabolites in Stabilizing and Protecting the Leaf Microbiome. This is a project that has been carrying out inside the Functional Metabolomics Lab, led by Daniel Petras, and EMN Lab, that is another group here in Tuebingen. So, and also in my case, there are many people that are helping, and without them, it will be not possible reach the place I arrived now with my data. Yeah, as you understand, I'm working with plant microbiome, and specifically I'm focusing on the phyllosphere. That is the include all the microbes that are hosted in the aerial part of the plant, and of course this microbe create interaction between themselves and between them and the plant. So as you can see here, let me see that where's the pointer? Pointer, yes. You see here a classical descriptive microbiome network, and you can see that there is in there we show in this network correlation between the microbial strain that are represented by nodes and the age represented the correlation. This correlation can be positive or negative. It means they are co-present in a specific moment, or they are absent when we do the metagenomics analysis of the plant in the case. But the aim of our job is mostly trying to decode the interaction that are between the microbes and the microbiome and understand what is behind this, trying to understand the mechanism or the function behind this correlation. And to do that, we use on-target metabolomics. I will talk mostly about siderophores. And siderophores, for who do not know, are small molecules or metabolites that are produced by both microbiome and the plant itself. And they are secreted in this apoplastic compartment, in the case of the plant, and they are finding the scarce element, that is the iron. And after that, these organisms are uptaking the siderophore, and it's like it's create an environment that is lack in iron, and actually protect the plant as well from possible phytopathogen. To study this interaction between the different microbes in the microbiome, I'm using a specific approach that is allow us to work in vitro condition, and specifically this we use the synthetic community. Synthetic community, for who do not know, are microbes that are isolated in a specific microbiome. They are assembled all together carefully, and then this allow us to understand what kind of small molecules are produced in a specific condition. And of course, to analyze the small molecules that can be shared between the strain, we use mass spectrometry and target metabolomics. The Stincone that I'm using, it's a Stincone that was assembled from a previous postdoc in the EMN lab. And the peculiarity of the Stincone is that we include 12 different bacteria strain and 3 different yeast strain. This is not common. I mean, to use two organisms that are coming from two different domains. And the specific syncom that we are using, it's representing the core microbiome. We saw this strain over the season, over the years. And inside this core microbiome, I'm focusing mostly on Pseudomonas. It's representing a keystone species in the Philosphere community, and Rhodotorola, that is well known as a strain that give protection to the plant. However, it's not known what is the impact of this micro yeast on the community, on the rest of the community in the microbiome. And we think that behind this interaction, there are siderophores that are mediating the interaction between the yeast and the bacteria. So to study the Philosphere, we are going with the Kemen lab. We are going to collect a plant in the field, wild plant, and in different season, in spring and fall, to catch different environmental conditions. And then after that, we came in the lab and we do DNA extraction, and then we do amplicon sequencing and data analysis. And from this data analysis, we get correlation network like this that it's possible to see here. In this case, thanks to the data that Mariam collected during her PhD, we were able to create this specific network in which we are using Rhodotorola as a main organism and are trying to understand what are the first correlated microbes that are observed along the 6 years of study in the field. And we saw that, interestingly, that between Rhodotorola, the yeast strain, and Pseudomonas, there is just positive correlation. But what is behind this positive correlation? And for that, we are using on-target metabolomics together with a dropout culture of the syncom. It's important here that you understand how it's done, this dropout co-culture. We have the syncom that include 15 different strains, as I said, and we co-culture all together, and we have the syncom co-culture. Besides that, we create 15 different co-culture in each one of these co-culture, we just dropped out one of the strain that are included in the syncom. These strain were grown on MM9 media agar plate, and after a few days, we have done metabolomic extraction, target metabolomics analysis, and molecular networking for molecular notation. With this approach, we are able to actually identify the single strain contribution in the full syncom metabolome. When we look at the data, the interesting pop up fast since the simple PCA, we have seen that when we drop out Pseudomonas from the co-culture of the syncom, we observe that there is a completely drastically change of the metabolome, as you can see here. While in all other case in which Pseudomonas actually is present and other strains are dropped out, we don't see all this difference like it's possible to observe here. And here it's much more clear in this heat map. You can see that when we dropped out Pseudomonas from the syncom, we have a metabolomic profile that is completely different in comparison with all the other conditions. And then you can see here that so we think that all this similarity is coming from the action actually of Pseudomonas in the community. And doing then, of course, feature-based molecular networking and looking for siderophore specifically, we figured out that rhodotoroic acid, that is one of, it's a siderophore and it's responsible for iron uptake in Rhodotorola, it was higher in amount when Pseudomonas was high out of the community. In all the other case, we see somehow a kind of stabilities on the concentration. Of course, when in the Stincone we drop out Rhodotorula, the product res out, and it means that the intensity is low. It's 0. So we start thinking, oh, but what is behind the Rhodotorula siderophore? What kind of other siderophore are present in the community? So here, thanks to the help of Prof. Allegraron, we create this query, and we applied on MassQL. That is a query language that has been developed by the GNPS group, and it allows us to ask a query and understand, figure out the presence of that kind of molecule that present a specific characteristic. In the case of the siderophore, we ask for molecules that are present in their apoform and the form in which the iron is bound. And beside that, and you can see here in this 2 part of the query, and beside that, the siderophore, the molecule that is binding the iron, need to have a specific isotope that is specific for the iron. So beside this in silico analysis, we have done also in vitro analysis. And here you see the classical native metabolomics approach that Daniel just mentioned. Instead of doing a protein infusion, in this case, we do a iron salt infusion. And then we do a metabolomics analysis. Of course, we run it 2 times, 1 time with iron infusion and 1 time without iron infusion, to then compare and perhaps see the shift in mass. From the MassQL query, what I got were a bunch of masses. And through anti-smash and GNPS, I was able to actually identify some of these masses. But what was much more interesting, and is related actually with the dropout experiment, is that we can actually define the importance in the community on the production of this kind of molecule. As you can see here, when we have a Stincone in which we drop out Pseudomonas, we have just 1 siderophore. We observed just 1 siderophore that actually I was able to identify as a Bacillobactin E that is produced by Bacillus. When Pseudomonas is in the community, we see that is in all this case, we see that the number of siderophore increase. So we can think also that Pseudomonas in the community is pushing other microbes to produce their own siderophores, because the scarcity of this element increase. So also the other members need to produce siderophore to scavenge the scarce iron present in the condition. And this was very, very nice. And then with the functional metabolomics approach, I was actually able to identify the main siderophores that I've identified also with MassQL. And you can see here Pseudobactin. Pseudobactin is the main siderophore produced by Pseudomonas. And interestingly, this siderophore was most of the case present in their binding form, the form in which is binding the iron. While the apoform was not actually observed also in the ion identity molecular network. And the reason why I assume is the fact that the apoform is present in a very low intensity, as you can see here. And this is becoming also, it can give information about the affinity that the siderophore has for the iron, in the specific case of the Pseudomonas. So in Pseudomonas have this siderophore with the with this high affinity for iron, other members need to produce, as I said, siderophores. And we see an high amount of rhodotorola, or rhodotoroic acid in this case, that is produced by rhodotorola, the yeast I'm studying, and also other yeast like this ferroxamine G that is produced by other members of the community. So, and with that, we start thinking that perhaps Pseudomonas is leading the community. And for leading the community, it's using also this high amount of siderophore produced by other members of the community. And this is a well-known activity that some, gram-negative positive, gram-negative bacteria have. And maybe this is behind this in-field data analysis, behind the interaction that Pseudomonas is using the siderophore synthesized by rhodotorola. And this is a well-known process, as I said, that is called the isosiderophore use. And we think that behind this positive correlation, there is actually the use of Pseudomonas of the siderophore synthesized by rhodotorola. But until now, we have just data from untarget metabolomics. And to confirm that, we start doing analysis growth curves and with all the members of the Stincone. And specifically, I grow all the members in both cases with fresh media, minimal media, and media in which we have 50% in volume of supernatant of rhodotorola. And interestingly, we observed that 6 members of the community actually, they are promoted by the presence of rhodotorola, or the rhodotorola isometabolome that is present in the supernatant. In all the other cases, we have no effect most of the case, and from Microbacter, you can see that there is an antagonistic effect. But until now, we have information about all the isometabolome of rhodotorola. And we didn't know at that point if it was exactly rhodotoroic acid that causes this, the siderophore produced by rhodotorola in high amount when Pseudomonas is there. So we fractionated the isometabolome or rhodotorola, and we, of course, identified the molecules present in each fraction. And by doing by activity assay, growth curve in this case, we observed that the fraction 3, that was the fraction carrying the rhodotoroic acid, presented an increase of growth compared with the control. And then beside that, we bought the standard for rhodotoroic acid, and we start doing growth curves to understand also the optimal concentration in which Pseudomonas is growing better than the normal condition. And then with this activity, we observed also that high amount of rhodotoroic acid, in the case of siderophore in general, have a negative effect on the growth of the strain. So, and this is, it's coming, I want to connect with the introduction. When there is a high amount of siderophores around in the microbiome, the member starts to actually suffer this lack of iron coming from coming off the scavenging or carried out by the siderophores. And that's why maybe some strain can get benefit just stealing siderophore of other members. With that, I would like to summarize all the findings from the in-field data, microbiome data. We observed that there is just positive correlation between the rhodotorola yeast and Pseudomonas strain. Then combining the Stincone with untarget metabolomics, we were able to see that there is actually a isosiderophore use by Pseudomonas for the siderophore produced by rhodotorola, and maybe this is behind this positive correlation. Actually, we are confirming that this is behind this positive correlation. With that, I would like to thank both Daniel Petras and EMN that are supervising me during this postdoctoral fellowship, and all the members of the Eric EMN lab and the functional metabolomics lab. And in case you have any question, I'm here to answer, perhaps.
Perfect. Thank you so much, Paolo. It was very fascinating. And big congratulations to both speakers. I mean, really, really fascinating lectures, both of you. We have also some questions in the Q&A. If you have any more questions in the audience, please drop them in the Q&A. But I have the great opportunity to ask as the first one. So I have some notes here. So I would like to ask Daniel about this micro spatter setup you have. I was just imagining the situation where it could happen that you would see this halo, so it means that you have some bioactive compound that is interacting with it, but you don't see any peak, for example. So, for example, I was asking in this case with the dynamic range maybe, or the concentration. Do you have problems with it, or how do you tackle this problem?
That's an interesting question. So you mean like we would not see any peak on the mass spec?
Yeah, or a very low one, let's say, or yeah.
I mean, that would be a great scenario. Typically, it's the other way around, right? We don't have a signal on the microspotter, as the mass spec is very much more sensitive. But then, I mean, maybe we would not use the right ionization mode. So we would maybe switch to negative mode, or make sure that we're like in the proper mass range. Right? So sometimes there's unexpected things that turn out to be bioactive. And obviously, with like ESI, we are biased towards a certain, range of like chemical space. So I would assume that might be the issue. Or, yeah. I mean, I'd hope that we still could ionize it somehow. But if you have no heteroatoms also in there, then probably you would need to switch to a different analytical technique.
Yeah. Then also I wanted to ask you, do you also switch columns in this setup? Do you use multiple columns?
We tried it with Hillig as well, yes. Because obviously polarity is sometimes an issue, and we're in one project we're particularly interested in very polar compounds. Technically, it works like the same. The only thing we're struggling with Hillig typically is like getting like very good reproducibility, and method development is a bit more challenging than with reversed phase. So reversed phase for us is simpler and more robust.
Yeah, maybe a last question regarding this microspatter setup, which is just interesting to me. So when you spot these droplets, does the composition of the mobile phase matter? Like considering the additives of the mobile phase, or composition generally if you use gradient evolution?
Yeah, it does. Not so much for the bioassay, because it's such a small volume that on the microfluidic device we dry it, right? So it's really completely evaporated before we do like the bioassay. But one thing that matters is like the high acetonitrile concentration at the end of the gradient. Then sometimes it overcomes the hydrophobic barrier in the microfluidic device. So we don't like to run the gradients to 100%, or rather than also use some makeup flow, to like bring it down, but more for like technical practicality of the microfluidics.
Okay, fantastic. Thanks a lot. We have a lot of questions now popping up in the Q&A, which is great. So maybe I can ask Paolo now one of these questions from the audience. So the question goes like this: did you perform any quantitative analysis of the metabolites? And if so, does the concentration range matter, or what did you observe?
So far, I didn't quantify any of just actually rhodotoroic acid, and it's present in a nanogram per milliliters.
Yeah.
But I didn't do target metabolomics for other molecules.
Also understood it was very exploratory, actually, so makes sense. Maybe one other question for you, Paolo, if it's okay. So it goes like this. Hi, Paolo, very nice talk. I agree. So are all the 15 Stincone members still alive at the end of the grow in co-cultures? And does the Pseudomonas or any other member tend to dominate? Because also it was my idea that the Pseudomonas is kind of a bully in this mix.
Yeah. So before to assemble Stincone, of course, there is a study that in which the person who assembled the Stincone study that all the members are fine together. We can detect them at the beginning and after incubation, their presence is there. And then there was a second, I believe, or not? It's just this. They are alive anyway. Yes, they must be alive.
Yes, there was also the question if the Pseudomonas is dominating. Yeah.
Of course, the Pseudomonas is dominating. When Pseudomonas is not there, there is another keystone species that take the place of it.
Yeah. Also, maybe just one more follow-up from my side. I was interested. So did you have to also optimize the media somehow to fit all these different types of microorganisms? And also maybe if you are focused on siderophores, is this media very low on metal ions to kind of let them try to catch for them? So kind of very
Yeah.
So I'm not a biologist, I'm sorry.
Yeah, no worry. We tried actually to somehow represent the plant condition. So we check the pH, and for the iron, it's a very minimal media. That's the condition that microbes actually can find in the leaf on any other microbiome, I think.
Okay, thanks a lot. Yeah, I will now just pass the baton to Loic. Here's some more questions from the audience.
Yes, we have more questions from the audience, and actually one of them, I had it as well. It is more common than a question. I think it's for, I mean, it can be for you both, but I think it was more for Daniel, and it's about the dark metabolome. So it's currently debating, and I think it was a work from Garisius that earlier this year that showed that there is no such thing as dark metabolome, or it's mostly artifacts. So what are your views on this dark metabolome?
Yeah, thanks a lot for this question. Very good one. Yeah, we've thought about this a lot since we saw that commentary from Gary. I think it's not a new observation. I guess like people since like a long time are aware that there's different ion adducts and that there's in-source fragments, and this is exactly where correlation analysis of MS1 data, such as with like various tools, tries to mitigate that. So there's RAMclust, there's a ton of other tools developed by the community, right? Our ion identity molecular networking is exactly built to mitigate that. And I think after clustering there, we still see like this large amounts of like unknowns, right? That we cannot like attribute to any in-source fragment. So I guess respectfully disagree on some points. I mean, I agree that there is in-source fragmentation, but I don't think that this is the majority of the signals we detect. And if you go from something simple like E. coli also to something like very complex, then that number just like gets bigger. I think there's been a very good blog post by Zamboni addressing that, and I think people in the community have very deliberate thoughts on that. There's a lot of technical details to that paper. I think while it's an elegant approach to reuse like existing data to estimate these things, I'm actually not 100% sure if they're talking about in-source fragmentation or fragmentation during ion routing, because of course they isolate at the beginning and have like MS data. So I'm not 100% sure whether that really reflects reality. And of course the paper made a lot of waves, but I wish the data would have been completely public to actually reproduce any of that analysis. But I assume there will be some more deliberate responses coming from the community.
Thanks. That was a tricky one. I think we can go for an easier one. It's a question from Rob Quinn from Michigan. So can you use your protein metabolite interaction method to discover novel protein metabolite interaction? It seems that you were using protein that you knew beforehand. Could you discover new protein in this way? I mean, I guess you answered pretty much, but...
Yeah, oh, well, good to hear that Rob is around. So yeah, that's a good question, and we've thought about that too. And we of course would love to use native mass spec to turn it around, right? To use like a full lysate of proteins and then figure out what's the binder. The problem is that resolution, so we tried that with size exclusion chromatography. It's not very good. So it's really hard to like separate a full proteome at the intact protein level. That's why we use a different technique in the lab. Actually, Paolo has been spearheading that as well to implement. So this is called thermal proteome profiling. This is developed by Mikhail Savitsky and others at EMBL in Heidelberg. And there you use like a proteomics technique to like have melting curves where you spike in like specific metabolites and then look at shifts and melting curves at the proteome scale to infer interactions. This has been working really well. There's a lot of work coming out. It's more in the proteomics field that this is applied, but there's I think like a lot of promise behind like these techniques. In parallel to that, there's also like limited proteolysis and some other approaches that people are using, and that's where I would put my money in. So I don't think native mass spec is fully there yet to resolve a full proteome.
Thanks. Another question on the bioactivity dereplication from Giselle Tamayo. So very interesting talk. However, to confirm the structure of an antibiotic, it is necessary to obtain larger quantity to verify the proposed structure with another orthogonal technique. Have you thought how to solve that unless it's coupled with the theoretical BGC?
Yes. Yeah. I mean, this is a very good question as well, and goes to the spot, right? So like there is still a bottleneck of like orthogonal structure elucidation for which NMR still is an elemental part, right? And you typically need to purify a compound, so that is really a remaining bottleneck. I of course wish there would be more streamlined approaches, maybe to deal with also online LC NMR couplings or some other, technical advances that could bypass that. On the computational side, I guess there's new tools emerging to facilitate the annotation of NMR spectra. SMART is one thing developed in the lab of Bill Gerwig and others. So I think there is progress, but I think the purification and the generation of NMR data is still like a key bottleneck. I see it mainly as a last step of like a long approach. So I think where our tools come in, they help you to accelerate like the pointing at the right peak, right? Because once you know what you're after, I think then it's like you can streamline the purification. And even though if you don't think about NMR, right, at the end you need to have the compound in hand anyway, right? So whether this is then through like purification or through total synthesis, or whether you can buy the compound if it's already commercially available, you really need to verify that. And then I think in all of the experiments we showed, like at the end we always either purified or bought the compound, such as for rhodoterolic acid, right, to really like make some follow-up hypothesis testing with this. And I think this is really important. Like a feature or a name, still like a pie in the sky. But what we want is we want to have like some verification at the end. I think that is really important. So yeah, if we could streamline that better to get more pure compounds faster, that would be great.
Yeah, and I had a question regarding the micro sampler system you created. So you said that with classical fractionation, you have like 30-second fraction and you can have like hundreds of compounds. So with this one, I guess you don't have like unique compounds in every well, right? So do you have a rough estimate of the number of compounds you have in your different microfractions?
Yeah. So this really depends on the complexity of the extract, right? So if we work with an environmental extract, with a biofilm or dissolved organic matter, or if we work with like a bacterial culture, right? Then that is of course like a huge range. But now for one of like the Pseudomonas strains we recently worked with, where we actually ID'd a new compound, which we purified then and got the structure by NMR. There at the end it was still a bunch of like overlapping ones, right? So maybe in the range of like 5 to 10. But then and that is the nice thing about like the correlation approach, you not just like say, well, they all overlap, but you actually start ranking, right? And you start with the one that has the best score. So you can point that one and then you can like go the row down and see. once you know them, you can also change the chromatography, right, to like get like some sort of like orthogonal information there. So it helps you to be a little bit more sure that this is actually like the bioactive.
Good. Another question from the audience. I have a question regarding the libraries used for spectral annotation. Do you rely solely on GNPS libraries available on the web server, or do you also upload specific libraries for your analysis?
Yeah, good question. So yeah, we mainly use the GMPS library. We did buy the latest NIST library, so this has been a great one. And then I know that there's massive efforts by the community, such as like MZMine, who are also like building new libraries, right? So there, yeah, I would advocate that everybody, you know, either uploads their data or provides standards to such like centralized approaches that we really can expand libraries. We did digitize the Tuebingen natural product collection. So thanks to Heike Protz-Österheld, we got like a bunch of like pure compounds that were purified over, I don't know, dozens, if not hundreds of PhD theses over the last decades. And yeah, we run them and put like all the MS information into the GMPS library. So that's our go-to solution. We don't have any in-house library that we keep for us. So we advocate for it and we do it. We just make it public. I think that's the way to go.
Thanks.
Well, thanks a lot. I'll give the mic back to Aleš, and I personally want to thank you both, Daniel and Paolo. It was very great to having you.
Thank you.
And now it's my time to shut my mic.
Thanks. I would just maybe also want to follow up on the last question about the annotations and libraries. I want to ask Paolo actually, how did you annotate these siderophores, or what, do you have any tips maybe also for?
From the MassQL result, yeah, annotated by using GNPS libraries, and then also I was checking in all the genome of the members and finding the best matching for siderophore in the case, and then I was doing manually, yeah, trying to understand fitting the mass with the iron, etc.
So when you have some really good candidate, do you also aim to buy the standard, let's say?
Yeah, of course.
Yeah, I'm just asking.
Yeah.
It's so intricate structures. I mean, how many standards are there? I mean, is it covered very well, or are there a lot of them? No, depends.
So sometimes they're hard to get, right? But in case of siderophores, we were very lucky that actually a person who commercially distributes some siderophores works in the very same building in Tübingen than we are. So we could just like walk down and buy it from them. It was a little bit expensive. But you know, when it's the last step and you have to pay like 200 euros or so, then it's okay. And I mean, here it's really I would stress again that we try to at the end up with authentic standards, not only to get level 1 IDs, but also to do some like follow-up experiments. And I think in the case of Paolo's project, it was really nice that, you know, after this different levels of phenotyping through the growth curves, at the end, you know, he could like nail it down and say, hey, here, if I add rhodotrolic acid, authentic standard, I see the same phenotype. So there we're like fairly sure now that this is like at least one of like the key compounds that drive the interaction.
Perfect. Thanks so much. As we are moving to the hour and a half, which we kind of have reserved, I think I will conclude with the last question. It's maybe open it a little bit more, because the question goes like this. So for protein-protein or protein small molecule interactions, could, for example, AlphaFold-like models provide some additional information to native MS? And maybe I would open it a little bit more. Are you using any AI tools for your data analysis right now, or are you testing it?
Yeah. I mean, there is already a lot of like AI or machine learning approaches that are broadly applied, right? Some of the supervised statistical tools we use, like Random Forest or XGBoost or so. But then also on the annotation side, like Sirius, right? So this is, I guess, a great example of an awesome tool that is available. For predicting interactions, we were very excited when the new AlphaFold version came that has now also some, small molecule interaction part. Unfortunately, that's mainly locked to a couple of few examples. So you can't just like put in any smiles, and then predict interaction. But I would assume that this will come, right? This is, I guess, their maybe business model. I'm not sure what the plan there is, but I assume they will lock this for drug discovery. But I would assume that also there will be some more open source approaches coming. And we're very much thinking about it. What I think is missing there is perhaps training data. While there is a lot of training data, thanks to, I don't know, decades of structural biology work for predicting structure, for interactions, there is less, right? So I think that is still something we can contribute to.
Perfect. Thank you so much. There are a few more questions, but I said we are kind of on the hour, and we have to end here. But we also provided during the registration, we provided your professional emails. So maybe if people can reach you to ask some additional questions, if that's okay, that would be great. And I would like to wrap up and thank you a lot. It was really fascinating lectures and kind of state of the art what is now in the terms of this microbiome communities and untargeted metabolomics in that case. Really amazing what is possible. Thank you so much for being here, for your valuable time. And as I said, this webinar is recorded and soon will be publicly available on the Metabolomics Society website and also on our YouTube channel. I dropped the links into the chat, but you can also keep an eye on our social media of the EMN. And thank you once again. And hopefully see you soon, maybe in Prague Metabolomics conference. We will see.
Thanks so much.
Thank you very much. Have
Thanks.
a nice day.
Ciao, ciao.
Bye-bye.
Bye-bye.